Namaste. Welcome to our poster. This is Shrivaishnavi Ranganathan, and my team and I are from the School of Chemical and Biotechnology at Shastra University, Tamil Nadu, India. The work we are presenting today is that of structural and functional mapping of the ARS operon in the bacterium Deinococcus indicus DR1. The ARS operon is responsible for providing resistance to arsenic in bacteria. It has two forms, ARS RBC in gram-positive bacteria and ARS RDABC in gram-negative bacteria. The operon codes for the following proteins: ARSR, a transacting repressor of the operon; ARSB, which is a transmembrane efflux pump that removes arsenite or AS3 plus ions from the cell; ARSA, an ATP-dependent protein that complexes with the ARSB; ARSC, an arsenate reductase enzyme that reduces arsenate or AS5 plus ions to arsenite ions; and an ARSD protein, which is a metallochaperone that collects arsenate ions from around the cell and brings it to the ARSAB complex. Deinococcus indicus DR1 is an extremophilic gram-negative bacterium that is isolated from the Dadri wetlands in India, and it shows very high resistance to arsenic. The proteins of its ARS RBC operon are yet to be characterized, and the mechanism adopted by these proteins together to remove arsenite has not yet been elucidated. The operon structure is as shown in figure 1. There is an arsenate reductase ARSC1 outside the operon at a distant location. The operon itself consists of 6 proteins: 2 ARSR repressors, ARSR1 and ARSR2, a metallophosphatase family protein or MPase as we have named it, 1 ARSB efflux pump, and 2 arsenate reductases, ARSC2 and ARSC3. The methodology we adopted is as follows. We first modeled the proteins, ARSB using the comparative modeling approach in the Rosetta server, and the other proteins using the deep learning approach called Rosetta Fold. A median model for further analyses was selected using the Thesis software. The models were validated using Ramachandran plots. Next, since ARSB is a membrane protein, its transmembrane residues were obtained using a hydropathy plot on the TM-HMM server. Subsequently, the CHAR membrane builder tool was used to build a lipid bilayer around ARSB. The latest top 6 membrane model was used as a reference for the lipid composition. After this, all the proteins were simulated in molecular dynamic simulations. ARSB was simulated in NEMD, while the rest were simulated in GROMACS, and the trajectories were analyzed. For all proteins excluding ARSB, the active sites were predicted on the CASP server, and their closest structural homologs were obtained from the DALI server. This step was followed by probing for the presence of structural equivalence among the predicted active site residues of our protein and the active site residues of the homologs. Lastly, multiple sequence alignment and phylogenetic analysis among the closest structural homologs were performed. Coming to the results, upon structure prediction, all structures were predicted as monomers, while ARSB was predicted to be a dimer. There are no known dimeric arsenic pumps recorded in literature, although other high-functioning heavy metal pumps that are dimeric do exist. The presence of less than 1% residues in the disallowed region of the Ramachandran plot is necessary to deem a structure as sterically stable and confident for further analysis. All proteins of the ARS operon in Deinococcus indicus DR1 had less than 1% residues in the disallowed region of the Ramachandran plot. Hence, we were confident of the predicted models. Figure 2 over here consists of 4 images with the MD trajectory analyses. The far left images are the root mean square deviation plots that describe the stability of the structure. The central images are that of root mean square fluctuation, which describe the flexibility of the residues. And the far right are images of the radius of gyration plots, which provide a measure of the compactness of the structure in the simulation. All plots plateaued much early into the simulation, with the latest being halfway through simulation time. This plateauing gave us confidence about the protein's performance in MD simulations. Figure 3 is that of the dimeric IRSB in a lipid membrane. The red region is that of the lipid bilayer, and the protein is in the center. The lipids in front of the protein have been sliced away for the sake of representation in this image. The wheatish color represents the predicted transmembrane residues. Figure 4 represents the results of active site prediction on the left and structural and superposition with top 10 structural homologs on the right. The pink highlighted region is the predicted active site pocket. Superposition with structural homologs was performed to check for structural equivalence among active site residues. We found that except for MPase, none of the proteins showed structural equivalence among the active site residues with those of the structural homologs. This prompts us to think that the proteins of the indicus DR1 utilize a different mechanism than that of the known structural homologs. Sequence and phylogenetic analysis showed considerable divergence from known structural homologs for all the proteins. To conclude our work so far, first, we see that while the proteins of the ARS operon in D indicus DR1 are structurally and functionally similar to their respective structural homologs, there is divergence on the sequence level and also at the efficiency of these proteins in tandem to confer resistance to arsenite. Second, the reason for ARSB being predicted as a dimer is unclear. We propose to probe further into the properties of these proteins and conduct systems biology analyses to elucidate the mechanism with which these proteins work together to make D indicus DR1 exhibit high levels of resistance to arsenic. Thank you for listening.